belongs to Latin and from approximately day. So they are actually in mammals represent all the temporal organization of the physiological processes that can occur in living organisms. They have different characteristics. For example, they have 24-hour cycle length approximately. They could be endogenously generated, and they could be even modulated by 24 hours environmental cycles. So they are, as we said, they are organized a particular in a particular internal system. In fact, the organism temporal function can be regulated by some internal mechanisms from a biological clock. As we can see, that is also called a pacemaker that's going to regulate and orchestrate the rhythms. So the stimuli belongs to an afferent pathway, also called input, and they are kept by some photoreceptor that have actually are very specialized for each stimuli. And from the pacemaker, they could be sent to from the afferent pathway, so the output going to the organs and peripheral tissues that going to express the rhythms. In particular, some rhythms parameters can be, as we said, influenced by some external environmental cycles. In particular, these environmental cues are also called synchronizers or environmental factors, or Zeitbergs from German, that means time giver.
Dr.
In
Francisca,
particular,
sorry to interrupt again. The slides are in number 2 only.
maybe it's better if I don't share the full screen.
Okay, no problem.
You, okay, maybe it's better so you can follow me.
Yeah.
So the most important and external environmental cues is the light-dark cycle, as you can see. But we also have even other external synchronizers, for example, the seasonal changes, behavioral, social behaviors, or feeding intake, or even exercise in our case. In particular, training could act as external synchronizers, as we said, altering and regulating the rhythmic characteristic of all physiological activities in mammals. So understanding the influence of the time of day on horses' physiological response to exercise and the mechanisms underlying the chrono performance could actually influence the management of training, the work routine, and even competitive events in horses. So the diurnal changes that act in the health-related physiological inflammation markers can influence the adaptation to exercise. So the aim of the present study was to assess the possible involvement of the circadian changes of certain physiological inflammatory biomarkers in the gain of exercise response in show jumping exercise. So the experimental part was divided into first step and the second step. So during the first step, the circadian rhythmicity and the inflammatory biomarkers has been assessed. We used the 10 Italian saddle horses that were maintained in boxes, and they belonged to the same training center and managed all equally. The thermal and hygrometric parameters were continuously monitored during this first step by means of a multiparametric probe, and the blood sampling test has been collected every 4 hours for a 24-hour period. While the second step aims to evaluate the interaction between the response to exercise and the existence of the daily rhythmicity of the previously investigated biomarkers. So in doing so, we performed actually an exercise schedule inside an indoor arena to get all the parameters to be standardized. So the thermal hygrometric monitoring has been performed with the same probe inside the indoor arena, and the exercise sections has been organized into steps in the morning at 10 a.m. and in the afternoon at 4 p.m. So during the exercise sections, the blood sampling test has been performed before the exercise, immediately after, and 1 hour after, following always the same show jumping training sections. So after the sampling test from the first and the second steps, the laboratory analysis has been performed. First of all, the hemochromocytometric exam and the blood smears has been performed for the evaluation of the leukocyte subpopulation. And the flow cytometry analysis has been performed for the identification of some lymphocyte subpopulation, in particular CD4 and CD8 subpopulation, by means of the same equipment. With the serum samples collected during the first and the second step, the enzyme-linked immunosorbent assay and ELISA analysis has been performed for the evaluation of the concentration of interleukin-1 beta 6 and the tumor necrosis factor alpha to evaluate the inflammatory response in horses. So first of all, the results of the first steps, as we can see, tells us the continuous recording of the ambient temperature, relative humidity, ventilation, and photo period, considering the 24-hour period conducted inside the boxes. And as we can see, all the parameters falling inside the thermoneutral zone considered for horses. So during the first step, the periodic phenomenon of each parameters that were analyzed have been evaluated by the application of a particular trigonometric statistical model to each time point. And this technique was also called the cosigner technique, that gives us some rhythmic parameters such as measure, amplitude, acrophase, and robustness of the rhythm. In particular, first of all, we analyzed the expression of circadian rhythms of the white blood cells, analyzed the total white blood cells. That gives us the idea to have an acrophase that falls inside the midnight time, so nocturnal rhythm observed with a high percentage of robustness. In the meantime, of all the leukocyte population, also only lymphocytes and neutrophils showed a daily rhythmicity with the same acrophase, so the peak of the rhythm considered during the early morning hours for lymphocytes and during the afternoon for neutrophils, as we can see, with the even high percentage of robustness. About the flow cytometry analysis, we evaluated the population of CD4 and CD8 lymphocytes, and we considered with the machine a pool of leukocytes previously selected with the machine that were focused and isolated to get the percentage of CD4 and CD8 positively marked cells with all the monoclonal antibodies. In particular, specifically within the selected areas, as we said previously, all the superficially marked cells for CD4 and CD8 were highlighted, as we can see from the graph, together with the nucleus to better distinguish the internal part from the external part of the cell that were differently marked with the different monoclonal antibodies. So in the specific analysis done with the statistical analysis, actually both of them exhibited diurnal rhythmicity. In particular, CD4 showed a peak of rhythm during the early afternoon hours, and the CD8 showed a nocturnal daily rhythmicity. For the serum sample analysis, we observed a daily rhythmicity for the interleukins, both interleukins, observed during the early morning hours and with a high percentage of robustness, and as opposite to the tumor necrosis factor, as we can see, that showed the peak of its rhythm observed during the afternoon hours, and this probably could be due to the fact that this parameters is also considered a good regulator of the sleep regulation in mammals. So it has the opposite trend of another sleep regulators, that is the the melatonin that is also known to have the peak of its rhythm during the nocturnal time. In particular, the second step of this present analysis showed has been performed with a two-way ANOVA for repeated measures to evaluate the significant effect of exercise and of the time of day, so during the morning and the afternoon exercise. In particular, we can see that actually the CD4 levels actually decreased during the afternoon hours after exercise compared to the exercise to the before values. So this could probably be an effect of rhythm, as we can see from the peak of its rhythm in particular. The same results could be seen for the CD8. In fact, it's actually going to rise during the afternoon hours compared to the morning hours. For the white blood cells, we can actually see probably the real effect of exercise during the morning, because we said that the white blood cells as a nocturnal daily rhythmicity. But in this graph, we can see that immediately after the exercise and 1 hour after exercise, we have a rise of the value compared to the before value, to the baseline values, only during the morning. So this could probably be done as an effect of the exercise. About the interleukins, we can see that they exhibited elevated levels during the afternoon compared to the morning hours. And this could probably be, as we said, from the CD4 and CD8, the effect of their daily rhythms. So in conclusion, the present chronophysiological investigation on the athletic horses showed that all the analyzed parameters actually show the daily rhythmicity considering the diurnal and nocturnal acrophases. So the inflammatory biomarkers did not show actually alteration due to exercise except for white blood cells, but probably due to the effect of the real rhythm. So the most of the effects were attributable to the rhythm of these inflammatory biomarkers. So the present findings actually could improve the understanding of the athletic horses' chronoperformances and trying to help the identification of the best time to get the training in horses. So that in the future, probably further investigation could be necessary to take into account maybe other inflammatory biomarkers, maybe, for example, the acute phase protein, or even considering other type of exercise applied, considering intensity, discipline, and so on. Thank you very much for the attention.